Well, hello, my fellow pre-meds, and welcome back to another video on metabolism. Today, we're going to be shifting our focus to lipid metabolism, a dense topic, no pun intended, but don't worry, we're going to break it down, digest it, convert it to ketone bodies, and send it right to your brain. So let's get started with an overview of fatty acid synthesis, which takes place in the cytoplasm of liver cells and fat cells, and to a certain extent, mammary glands as well. So what's going on over here is that you have these two carbon building blocks, two carbon molecules known as acetyl-CoA being joined to one another until you get a 16 carbon fatty acid known as palmitate. So you can see that right here. This is the structure of acetyl-CoA. What we're really interested in is this acetyl part over here. The rest is just extra baggage. It's not going to be incorporated into the fatty acid chain. And you have 7 rounds, even though you're joining 8 acetyl-CoA groups to one another. And you can see why over here. So let's say this is the first one. This is the first round of addition, second round, third round, fourth round, fifth round, sixth round, and seventh round. So this is a 16 carbon molecule. We only require 7 rounds of adding acetyl-CoA. And you can see that that's why you only have 7 ATPs being consumed and 14 NADPHs, because each round is going to require 1 ATP and 2 NADPHs. And of course, it's stimulated, this process, by insulin. And that makes sense. If you're in the fed state after you just had a meal, you secrete insulin to store away some of the excess fat that you just got from that meal. And then, of course, it's inhibited by glucagon and epinephrine. All right, but we do run into a problem because, as you may know, acetyl-CoA primarily exists within the mitochondria, within the mitochondrial matrix. But fatty acid synthesis takes place in the cytoplasm. And because acetyl-CoA is this large molecule over here, it can't freely cross the membrane and move across different compartments. So the solution is that you want acetyl-CoA to condense with oxaloacetate, which recall is the first step in the Krebs cycle, to form citrate. Then citrate, via the citrate shuttle system, can cross into the cytoplasm and be converted once again by citrate lyase into acetyl-CoA and oxaloacetate, so that acetyl-CoA can feed into fatty acid synthesis. Now, of course, elevated levels of citrate will inhibit the Krebs cycle, and that makes sense because if you have a lot of citrate, that probably means that the cell is energetically satisfied. So the Krebs cycle can probably stop. We don't need more energy, and we can shift that citrate, convert it to acetyl-CoA, and allow it to synthesize fatty acids. Okay, so now let's go over the individual steps of fatty acid synthesis. So the very first step, carried out by acetyl-CoA carboxylase, which is the rate-limiting enzyme. As the name implies, carboxylases acetyl-CoA, adds a carbon atom to it, and now you have this 3-carbon molecule known as malonyl-CoA. Notice it requires biotin and ATP and bicarb, just that carbon atom, to carry out this reaction. Now, after that, we have another enzyme that carries out a series of reactions. This enzyme is known as fatty acid synthase, carries out these 4 reactions to build the growing fatty acid chain. And rather than reading these for you, I'm going to show you how this process takes place. So you can see right here, we're starting off with our acetyl-CoA molecule, and then we add this malonyl-CoA, this 3-carbon molecule. Now, this is very important. You're not adding acetyl-CoA every cycle. What's happening is you're adding this 3-carbon molecule. So technically, this is just the growing fatty acid chain. So right now it's 2, but in the next cycle, this one will be a 4-carbon molecule. And in the next cycle, it'll be a 6-carbon molecule. But if we're adding this 3-carbon malonyl-CoA molecule, why is it only adding 2 carbon atoms per cycle or per round? Well, that's because you're releasing one as CO2, 1 carbon atom as CO2. So does it make sense? Like you added a carbon atom over there just so that you release it over here? Well, it turns out that this is sort of a thermodynamic trick where the body or the cell stores some excess energy in the form of that carbon atom, and then in order for this to be more energetically favorable, it releases that carbon atom and then incorporates only 2 carbons in the growing chain. So now you can see that this yellow carbon, this carbonyl is right here, the black one is over here, and now you can see this carbon atom is attached to the black carbon, and then the red one is attached to the green one, and the purple one has been given off as CO2. So this step is called the condensation step. The next step is known as the first reduction step. So you see we use NADPH to reduce. What are we reducing? We're reducing a ketone. When you reduce a ketone, what do you get? You get an alcohol, specifically a secondary alcohol, as you can see right here. So the step after that is going to be a dehydration step to essentially get rid of this OH, get rid of this alcohol. When you dehydrate an alcohol, you end up with an alkene. So you can see right here, now we have an alkene. The final step in the synthesis of this growing fatty acid chain, or in the first cycle of this growing fatty acid chain, is going to be to saturate this double bond to form a single bond. And the way we saturate it is via a reduction step using NADPH. So now you can see we have a 4-carbon molecule. You can see you have the yellow, black, green, red, as we had right here, yellow, black, green, red. Now, importantly, this molecule, this four carbon molecule, in the next step is going to be in this position, and then the next molecule we're going to add is another malonyl-CoA. So you're not adding another acetyl-CoA. Hope that's clear. Okay, now after you've made fatty acids, in order to store them away in the body, you want to store them in the form of triacylglycerols. This is pretty straightforward. You just activate them, adding that CoA to them. So now you can see you have a fatty acyl-CoA once it's in this form, and then it undergoes a reaction. So 3 of them will undergo a reaction with glycerol, and then you get triacylglycerol, hence the name, acyl. So these are 3 acyl groups, so triacyl, and then glycerol, that 1 glycerol molecule. Pretty straightforward, and more on this in our lipid structure and function slide deck that we released earlier in this playlist. Okay, now let's talk about fatty acid breakdown. So now we've built fatty acids, but in the future we may need to break them down to derive energy from them. So let's talk about the breakdown of fatty acids. So the first step before you can break down a fatty acid, you need to activate it. And the way you activate it is what we just saw, like by adding CoA to it to make it fatty acyl-CoA. So it turns out that there are different kinds of fatty acids, like based on the number of carbon atoms they have. So you have short chain fatty acids, they have like 4 to 6 carbon molecules, 4 to 6 carbon atoms. Medium chain have around 6 to 12, and then long chain fatty acids have 12 to 20 carbons, and then finally very long chain have more than 20 carbon atoms. So all of them, with the exception of the very long chain fatty acids, are metabolized or broken down in the mitochondria. The very long chain fatty acids start off in peroxisomes. Now, importantly, the short and the medium chain fatty acids can just easily diffuse across the membrane, but the long chain fatty acids, they need to be carried into the mitochondria for breakdown via this shuttle system known as the carnitine shuttle system. So we've already activated them using fatty acid plus CoA plus ATP to form fatty acyl-CoA. But and notice we used energy over there, but we're breaking fatty acids to be able to generate more energy. So presumably we're going to be netting more afterwards. Like at some point we're going to make more than just 1 ATP, and that's why it turns out to be energetically favorable. Okay, so it's transported into the mitochondria after it's been activated by this carnitine shuttle system, essentially where you get the fatty acyl-CoA and you just replace that CoA with this carnitine molecule, okay, using an enzyme known as carnitine acyltransferase 1, or CAT1, which is the rate-limiting step of this whole process. So once you have the fatty acyl-CoA in the form of fatty acyl-carnitine, then the carnitine shuttle system can transport it into the mitochondria. Once you're inside the mitochondria, carnitine acyltransferase 2 will transfer the carnitine off the acyl group and add CoA once again, and then you can carry out beta-oxidation, which we're going to go over the steps right now. And they're essentially the reverse steps of fatty acid synthesis. So if you understood fatty acid synthesis, you're probably already going to know this, but you'll see right now. All right, so this is fatty acid synthesis. These are steps of fatty acid synthesis that we just saw. Now let's talk about the steps of fatty acid breakdown, or fatty acid beta-oxidation. So first of all, why do we call it beta-oxidation? Well, you can see right here, this is the carbonyl carbon. The one adjacent to it is the alpha carbon, and the one adjacent to that is going to be your beta carbon. And because the first step over here is oxidizing this beta carbon, we call it beta oxidation. So you can see right here, we get FADH2. So the idea there is that you're adding to this FAD plus. FAD plus is being reduced into FADH2, and this is being oxidized, right? Because FAD plus is an oxidizing agent. So now you have your double bond over there because you just oxidized your single carbon-carbon bond. After that, you're gonna hydrate. So again, just reversing that process. Now you're adding water rather than dehydrating. When you hydrate a double bond, an alkene, you get an alcohol, as you can see right there. After that, the next step, as you may have guessed, is again going to be another oxidation step to oxidize the alcohol into a ketone. And once again, we needed an oxidizing agent, so we used NAD+, and that's why we're releasing NADH as part of this process. And remember, NADH and FADH2 can eventually participate in the electron transport chain and generate ATP for us. So that's part of this fatty acid breakdown and generating energy over there. Okay, now the last step is going to be to break this bond. So, we have this alpha, beta, the bond between the alpha, beta carbons right here. We're going to break this bond in order to release an acetyl-CoA molecule. And again, that can also participate in the Krebs cycle and generate more ATP for you. So you can see right here, and I really want to clarify this. So what's going on here is in this process, you're adding coenzyme A. You can see that this coenzyme A is now part of this growing or shortening fatty acid chain. So you can see it was added right here where you broke the bond between the alpha and the beta carbons. So now this coenzyme A will be attached to the beta carbon and what's after it. So you're going to have more carbons. Of course, this is just a 4-carbon molecule. This originated as a 4-carbon molecule. So you can see that you have only two carbon molecules. But if this were like 6 carbons long, then you would have a 4-carbon molecule over here. And then this one that you just released is going to be your acetyl-CoA. Hope that makes sense, okay? So the carbonyl carbon and the alpha carbon of your original fatty acid chain are what are given off as acetyl-CoA. And then the other part, the remainder of it, is going to be your shortening chain, okay? Now let us move on to regulation and yield of fatty acid metabolism, the energy yield, that is. So the regulation is activated by glucagon and epinephrine. This makes sense. Epinephrine is released in the fight or flight state or even just in the fasting state, so you're going to need to break down some of those fatty acid molecules, and then insulin will shut the process down. Now importantly, malonyl-CoA inhibits CAT1 so as not to have fatty acid oxidation and synthesis running simultaneously. What does this mean? So let's go back a few slides over here, right here, where we mentioned that the first outcome, the first product of the first reaction in fatty acid synthesis is this malonyl-CoA molecule. And remember that this was the rate-limiting enzyme over here. So when you have a lot of malonyl-CoA, what does this mean? This means that I want to synthesize more fatty acids. Probably means that insulin is being released so I can synthesize more fatty acids. Well, then this malonyl-CoA molecule will go to carnitine acyltransferase, which is the rate-limiting enzyme of fatty acid oxidation, and inhibit it. So malonyl-CoA, the outcome of the rate-limiting enzyme of fatty acid synthesis, will inhibit the rate-limiting enzyme in fatty acid oxidation. And this makes sense because fatty acid oxidation and synthesis are opposite reactions, so they generate the opposite of one another. So you don't want them running at the same time. It would be futile to be synthesizing fatty acids and simultaneously carrying out beta oxidation to break down those fatty acids. That would be a waste of energy and resources. So that's why malonyl-CoA will inhibit the carnitine acyltransferase enzyme. All right, let's get back to our other view over here. Okay, so when it comes to energy yield, you get a whopping 104 ATPs per palmitate molecule. Again, feel like I'm selling my product to Shark Tank over here, but these are the numbers we have. So 8 acetyl-CoAs plus 7 NADHs plus 7 FADH2s. These are the outcome of breaking down one palmitate 16-carbon molecule. So then, and I just want to make it clear why you get 8 acetyl-CoAs, but you're only getting 7 NADHs and 7 FADH2s. And that's because if you take a look over here, this is our 16-carbon skeleton, okay? If you were to break down this first one, so there's one round of breaking, 2, 3, 4, 5, 6, and then that very last one, when you break down number 7, you're going to get two carbon molecules over there. So that's why you end up with 8 acetyl-CoAs, but you're only doing it 7 times, so you get 7 NADHs and 7 FADH2s. And then we know that each acetyl-CoA will give you 10 ATPs once it enters into the Krebs cycle, so it's going to give you 3 NADHs, 1 FADH2 and 1 GTP, which is equivalent to ATP. So if you do the math, you get 10 ATPs per acetyl-CoA. And then finally, if you do all of the math over here, you're going to get 106 ATPs. But remember, we mentioned that you need ATP to activate this whole process. So remember what we said, when you want to add the coenzyme A to the fatty acid to start the process, you need to add ATP. So that's where you lose 2 ATPs. So you net 104 ATP molecules per palmitate. All right, now we're going to shift our attention to ketone bodies. So I'm going to start with a broad overview, and I really want to emphasize that when it comes to ketone bodies on the MCAT, it's more important that you understand the broad picture, why they're used, how they're broken down, rather than understanding the detailed steps of synthesis and breakdown. Okay, so let's go over the broad picture. So up until now, we've been talking about fatty acid synthesis and breakdown. And it turns out that the largest reserve of energy in our bodies is in the form of fat. But it turns out that unfortunately, because these fatty acids are really large molecules, they can't cross freely across membranes and into different compartments. And even if you were to break them into acetyl-CoA molecules, again, those acetyl-CoA molecules contain that large CoA molecule. So like the fatty acids, they can't just simply cross membranes and move across different compartments. So the body's found this solution where first you break the fatty acid into acetyl-CoA, and then the liver cells can convert acetyl-CoA, this large molecule over here, into smaller molecules known as ketone bodies. So we have acetoacetate, beta-hydroxybutyrate, and acetone. Notice that they're not necessarily all ketones. So like this one doesn't even contain a ketone, but that's just their name. Their name is ketone bodies. And then these ketone bodies can be transported out of the liver into different compartments within the body, in the brain, the skeletal muscle, et cetera, et cetera. And then once they're in their desired location, their desired compartment, they can then be converted back into acetyl-CoA and used for energy in the Krebs cycle. Okay, so that's a broad overview. Again, I'm going to go over the problem they solve, but put it in context. Okay, so fatty acids and acetyl-CoA cannot cross into the blood-brain barrier. You might say, well, this isn't really a big deal. The brain can just use glucose, right? But what happens in periods of fasting or intense starvation? What's going to happen is first, you're going to rely on the first few hours, you're going to rely on the glucose that you just had from your meal, so the free glucose in your blood. When that runs out, you start to rely on glycogen. When those stores run out, let's say about 12 hours later, now you need to start relying on gluconeogenesis, right? Now, the problem with gluconeogenesis is that it relies on certain raw material, and one of those raw material is from amino acids, right? You break down proteins, you break down muscle to generate these amino acids to be able to carry out gluconeogenesis. So it's probably not the wisest idea to rely solely on gluconeogenesis. Add to that the fact that you have these large deposits of fat just stored away there. So that's why the body figured out this way of being able to package the acetyl-CoA in the form of ketone bodies to ship them to the brain and different compartments of the body to be able to use them as energy sources so as to decrease the reliance on gluconeogenesis and decrease the breakdown of proteins. So the timeline over here, around 12 to 24 hours, is when you start gluconeogenesis, but at the same time you'll start the process of fatty acid breakdown, and the acetyl-CoA over there will be shunted away towards ketone body synthesis, and we'll see how that works in just a bit, but it's shunted towards ketone body synthesis. However, early on, around like the 24-hour mark, all the ketone bodies you're making are being taken up by skeletal and cardiac muscles, because it turns out that these muscles are just so good at using those ketone bodies, so there's nothing left for the brain. So at this point, the brain is still relying on gluconeogenesis for its energy. However, as you continue to starve, 72 hours plus, the brain is not going to want to continue to use glucose because now it's breaking proteins to use the amino acids for gluconeogenesis. So at this point, the body starts to make more and more ketone bodies to the point where no matter how many ketone bodies the skeletal and cardiac muscles use, there's always more left over for the brain. And at that point, 75% of your brain's energy is coming from ketone bodies to spare the breakdown of proteins to synthesize glucose via gluconeogenesis. All right, so here's a big picture of how ketones are synthesized. Essentially, ketone bodies are synthesized in the mitochondria of liver cells from acetyl-CoA to ketone bodies. There's a lot of steps there. The rate-limiting enzyme, you should memorize the rate-limiting enzyme, is HMG-CoA synthase. But what triggers the synthesis of ketone bodies? Because if you've just had a meal, you're not necessarily making ketone bodies because you're actually storing away the fatty acids. However, in a period of fasting, when insulin levels fall, glucagon and epinephrine will start to rise, what's going to happen is you are going to start synthesizing glucose via gluconeogenesis because you're fasting, because let's say it's at 12-hour mark, okay? Oxaloacetate is being depleted because oxaloacetate turns out to be one of the precursors for gluconeogenesis, as we discussed in our first part of our carbohydrate metabolism video. So once oxaloacetate is depleted, but at the same time, you're making more and more acetyl-CoA because you're breaking down fatty acids. What's going to happen in this case? Well, let's take a look at the Krebs cycle. The Krebs cycle relied on acetyl-CoA joining together with oxaloacetate to form citrate and carry out the cycle. Well, because oxaloacetate is being shunted via Pepsi-K to form phosphoenolpyruvate, which recall was an intermediate of glycolysis, to essentially reverse the process of glycolysis and perform gluconeogenesis. Now we have little oxaloacetate that is not able to bind all of the acetyl-CoA. So instead, the Krebs cycle is not going to be running. Rather, acetyl-CoA is going to be shunted to ketone body synthesis. So the idea is that when a cell halts its Krebs cycle because it's performing gluconeogenesis, it is going to start shunting acetyl-CoA to ketogenesis. And that's exactly what happens in liver cells, and only in liver cells. Okay, so the regulation is pretty straightforward. If you have high acetyl-CoA, it makes sense that you're going to shunt some of it to ketone bodies. If you have high NADH, recall from our video on the Krebs cycle, we said that NADH, because it's a product of the Krebs cycle, is an allosteric inhibitor of the Krebs cycle. So it's going to shut the Krebs cycle and activate the synthesis of ketogenesis. Glucagon, so let's think of it in terms of insulin. We just mentioned that insulin is involved in the process of storing away fat, so it's going to inhibit ketogenesis, right? Likewise, glucose. Insulin is released when you have a lot of glucose. So if you have a lot of glucose, you're going to shut down the process of ketogenesis. It also makes sense. Like if I just ate a meal and I have a lot of glucose in my blood, I don't need ketone bodies to use it for my energy because I have glucose available, right? And then the reverse of insulin, if you have glucagon, then it's just going to activate the process of ketogenesis. All right, so now these are the individual steps of ketogenesis. Again, I don't think they're very high yield. I would know that HMG-CoA synthase is the rate-limiting enzyme, and then I would just understand that it goes from joining together two acetyl-CoA molecules to eventually giving you these ketone bodies. Of course, our body will only use acetoacetate and beta-hydroxybutyrate for energy. Acetone is not used for energy, so it's not converted back to acetyl-CoA in the process of ketolysis, as we'll see in a bit. All right, here's just a tie-in to diabetic ketoacidosis, which is one of the complications that occurs with type 1 diabetes mellitus primarily. So you don't need to know the clinical aspects of diabetic ketoacidosis or diabetes for that matter for the MCAT. A lot more on that in med school, but I think this is relevant to the biochemistry of it all, so I think it's worth knowing. So essentially, when in the case of type 1 diabetes, your body isn't able to generate insulin or secrete insulin, and that's why people have to take their own insulin exogenously. And so usually, like you'll have a child, because type 1 diabetes mellitus is more common in children, that forgot to take their insulin dosage. So now what happens if you don't have insulin? You're unable to store away fats, and your body starts to uncontrollably break fatty acids, and then the acetyl-CoA from those broken fatty acids will be shunted towards ketogenesis. Ketogenesis, because the ketones are acidic, will cause metabolic acidosis. That's just a fancy way to say that the blood is more acidic. And then what happens is that you'll see these patients will start to hyperventilate or deeply ventilate, which we call Kussmaul breathing. And the reason they're doing that, and this is a tie-in into a high-yield concept on the biology section of the MCAT, when you hyperventilate, you're blowing away CO2. And when you blow away CO2, you are essentially alkalinizing your blood. So you're making the pH higher. And that makes sense. They're trying to compensate for the lower pH that was caused by those ketone bodies. And just a fun little fact over here, if you smell their breath, you'll smell it's a fruity breath, and that's because acetone over here has that fruity odor to it. Okay, now let's talk about ketolysis. So ketolysis occurs in peripheral tissue such as the skeletal, cardiac muscles, also occurs in the brain and the kidneys. Importantly, It turns out that ketolysis is mostly occurs in tissues that are metabolically active. And this makes sense. If we have tissues that are very metabolically active, like they need a lot of energy, in periods of intense starvation, the primary source of energy is going to be ketone bodies. So it makes sense that those tissues are going to be using most of the ketone bodies. And it actually turns out that the liver can't use any ketone bodies. And here's why. So if you look at the process of breaking down ketone bodies, so now we're going from beta-hydroxybutyrate or acetoacetate, which were the two ketone bodies that our body uses, and then converting them back to acetyl-CoA so that the metabolically active tissues can use that acetyl-CoA for energy in the Krebs cycle and the electron transport chain. The rate-limiting enzyme over here is succinyl-CoA transferase. If succinyl-CoA sounds familiar to you, that's because it's one of the intermediates of the Krebs cycle. So this means that in order for me to carry out ketolysis, I need the cell to have an active Krebs cycle. And we mentioned that in the liver, when we're trying to make ketone bodies, we have to shut down the Krebs cycle. So if the Krebs cycle is shut down in the liver, then that means the liver is not going to be able to break down the ketone bodies that it just made, and that makes absolute sense. You don't want to make and break ketone bodies in the same compartment. Rather, you want to synthesize those ketone bodies, send them off in the bloodstream to other organ systems that are metabolically active that do have succinyl-CoA because their Krebs cycles are still active and therefore can break down the ketone bodies to acetyl-CoA and use that as energy source. Another reason why the liver can't carry out ketolysis is because it actually lacks this enzyme, succinyl-CoA transferase. Okay, now let's move on to cholesterol. Well, before I move on to cholesterol synthesis, I want to do kind of a quick recap on what this whole problem of ketone bodies, like what problem did ketone bodies solve? So the body has this problem. Its largest source of fuel is stored in the form of fat, but it can't transport the fat or the product that comes from breaking fat, which is acetyl-CoA, to other parts of the body because they're highly compartmentalized, they're large molecules, they can't really cross membranes, they can't move across different compartments. So you might say, well, not a big deal, we'll just rely on glucose for transporting energy sources because it's a lot smaller, a lot easier to move around. So early on, after you've just eaten a meal, you're going to use the glucose, the free glucose in your bloodstream. After that, you're going to use glycogen. Once that's done, you're going to start doing gluconeogenesis. Well, now is when we really run into the problem, because if you're using gluconeogenesis, at some point, you're going to start breaking your proteins to use amino acids in the process of gluconeogenesis, and of course, we do not want that. So the body came with this clever way, the liver in particular, came up with this clever way where we break fatty acids, acetyl-CoA, and then package the acetyl-CoA in the form of ketone bodies to be able to ship those ketone bodies out of the mitochondria and into different tissue other than the liver. And then once the ketone bodies are in their desired locations in the brain and the skeletal, cardiac muscles, they can then be converted back into acetyl-CoA and undergo the process of ketolysis. Okay, let's move on to cholesterol synthesis. This is pretty straightforward. I don't want to spend too much time on it because it's not very high yield on the MCAT. What you want to know is that cholesterol is synthesized from acetyl-CoA, so it's converted to HMG-CoA. Remember, HMG-CoA was also involved in the synthesis of ketone bodies. HMG-CoA synthase in particular was the rate-limiting step in ketone body synthesis. Well, over here we have HMG reductase as the rate-limiting step of cholesterol synthesis. But importantly, and again, this ties to the idea of compartmentalization, ketone bodies are made in the mitochondria, and that's where you're going to find HMG synthase. Whereas HMG reductase is found in the cytoplasm, which is where cholesterol synthesis takes place. And again, you need NADPH, that reducing agent to synthesize cholesterols. And just a tie-in into our lipid structure and function video, remember those isoprenes that we said are the building blocks of cholesterols? They look like that. Sometimes they'll show up on the MCAT, so just know that as well. Okay, and then one important thing I forgot to mention is that they not only occur in the cytoplasm, but also in the smooth endoplasmic reticulum, more on the different parts of the cell in our biology playlist. Okay, these are some knowledge checks. I just speed through these, but if you have any questions, just reach out to me and I'll be happy to provide you with explanations over here. Okay. All right. Well, that was the end of our discussion on lipid metabolism. Again, a very dense topic. I hope that you really try to understand the big picture, especially when it comes to ketone bodies, synthesis, breakdown, and why the body makes them, because those are a lot more important than understanding, which enzyme carries out which reaction when it comes to the breakdown of ketone bodies. So if you feel like you need to watch the video again, watch it again. If you have any questions for me, reach out to me. Use our Anki cards to aid with memorization, and I'll see you in the next video.